Today we're going to talk about viruses. So viruses are not really classified as living things, basically because they cannot really reproduce themselves. And as we'll see down here, there was a weird crystallization thing that typically we don't throw them in the living thing category. So what does that make them then? Well, it kind of depends a little bit on the history as we go through. So in the late 1800s, scientists were kind of looking for the cure for tobacco mosaic disease. And we kind of have to understand the anatomy and physiology of a tobacco plant to kind of see why this was a big deal. So tobacco plants, as we can see up here, we use the leafy part of the plant. And if we have the tobacco mosaic disease, was kind of giving these little splotchy spots on the plant. So as a farmer takes the leaves and dries the tobacco, splotchy part would not be good. And so this was a big deal back in the 1800s because a lot of the southern economy and kind of the outlying economy depended on tobacco as kind of a cash crop. And so it started with a guy by the name of Dmitry Ivanovsky, and he kind of passed some sap of infected plants through filters that could handle bacteria. We knew about bacteria at that point in 1892, but after it passed through the filters, the new leaves were still infected. And so that tells us, wow, this stuff is really, really tiny, whatever is causing this disease. And the fact that it's causing as a disease would be like a pathogen. Next we had a guy by the name of Martinus Badrinac just a few years later. And he was passing in the infected sap through agar gel, and still the infection would be passed along to healthy plants. And so he kind of is credited with coining the name virus, which means Latin for poison. And it wasn't until about 35, 40 years later that a guy by the name of Wendell Stanley then purified and crystallized this tobacco mosaic disease or virus, which is not really what living cells can do. They don't really crystallize that well, and so that's why we kind of don't qualify them as living, in addition to them not being able to reproduce themselves. But he had to wait until the electron microscope really kind of came into full swing that allowed him to see the viruses and the crystallization aspect of it. And so that's kind of the history of how we started with our virus science. We now know that a virus has kind of its anatomy, a protein coat called a capsid on the outside. Sometimes that capsid then is surrounded by an envelope, and that helps it infect the host cell. And then on the inside, it's going to either be DNA or RNA as the nucleic material. And so the virus itself can't really infect, or it can't really reproduce itself, so it has to infect a host and then kind of trick that host into making copies of itself. And so there's lots of different shapes of virus capsids. So here's the classic T4 bacteriophage, kind of what Hershey and Chase were studying. Here would be tobacco mosaic. It's more like a spiral spring. And then here would be kind of the influenza virus, which is kind of this spiky ball kind of shape. And all these shapes then allow them to infect the host cell that it specializes in infecting. So we as humans probably wouldn't be too susceptible to this bacteriophage because it doesn't specialize in that. But as far as the influenza virus, it does very well against us as humans. So the two cycles then that we have are the lytic and the lysogenic cycle. And so we'll start with the lysogenic. It's definitely a little less harmful. So the infection takes place, the DNA gets put into the host chromosome, and then in the lysogenic, that DNA just kind of hangs out in the host DNA. And so every time mitosis takes place and the cell gets copied, the virus genome gets copied as well. And so that might last for months, it might last for years, it might last for decades until some sort of environmental trigger then would cause it to exit the lysogenic cycle and then go into the lytic cycle. That could happen, or a virus could infect the host cell, and then it could just do the lytic cycle all the time. So this infection then would take place. We would have kind of an incubation period. So most viruses incubate. Something like chickenpox might be about 7 days. Something like the flu, maybe a day or so. Something like HIV, maybe up to 7 years. And so that incubation will take place. All the while, copies are being made by the host unsuspectingly. And then when the time is right, the viruses will lyse out of the host cells and then will attack the host organism. And so that's the lytic cycle, and that is definitely more harmful to the host as there's a direct attack on the host. We have different incubations, as we see, of different viruses, and our main weapon against that then would be vaccines. So viruses, your body has to fight it off with its own immune system. And so we can't really give you antibiotics. We can kind of treat the symptoms, but we can't really treat the actual virus itself. And so that's why antibiotics don't work. That's why your immune system has to do the heavy lifting. And so a vaccine, the idea is we're going to give you a kind of a wimpy version of the virus that will give your body's immune system kind of the blueprint to fight that off. And then when that happens, then ever after, hopefully, you will be able to take on that strain of virus and be able to fend that off without any problem. So that's a little bit about virus, kind of the overview of how they work, and we'll kind of get into some specific viruses in a few different slides, but I hope that was helpful.